Hello everyone and welcome back to another MCAT ACE lecture. In today's lecture we are going to discuss the eukaryotes, prokaryotes and viruses. So since this is kind of like a long lecture, we are going to divide it into two sections. In the first video or part 1, we are going to discuss cell theory and then focus more on prokaryotes, including different modes of classification for prokaryotes, and then go over structure and their genetics. And then in the second lecture, we will go over eukaryotic cells, describe the function of different organelles, cytoskeleton, and then finally go over the mechanism of replication of viruses, prions, and viroids, and then as always conclude our lecture with some practice questions. Alright, so starting with the first part of this lecture, let's start with the cell theory. So the first time that the cell was discovered was in the 17th century, and that was when Robert Hooke has observed a cork, a slice of cork, under a compound microscope. All he could see was a multitude of tiny pores that resembled kind of like compartments of a honeycomb. And from there on, he gave the name cell to these structures. But then, because a cork was kind of like a dead structure, he couldn't see the internal structures of a living cell. It was like about 10 years later that scientists were able to see a live cell under a microscope, and they described cells as moving organisms. So the cell theory basically refers to the idea that all living organisms are made of cells. So all living organisms are made of cells. Then the second part of cell theory tells us that the cells are the basic building unit of life. So cells are the basic building unit of life. And then the third point is that new cells arise from pre-existing cells that divide in two. So new cells arise from pre-existing cells that divide into two. Okay, now these first 3 points are referred to as classic cell theory, but then over time, when there has been advances in genetics and molecular biology techniques, new tenets have been added to the cell theory. So therefore, the modern cell theory has added these new points, which include that cells carry hereditary information that are passed from parents to daughter cells during every division. So cells carry hereditary information, like for instance DNA, that is passed to daughter cells in every division. The next point is that some organisms are made of only one cell, while others are made of multiple cells. And so these are unicellular versus multicellular organisms. And then finally, modern cell theory states that there is energy flow within cells. So there is energy flow, or in other words, there is metabolism that is happening inside cells. Okay, now there are some exceptions to the cell theory, and these include, for instance, mitochondria and chloroplasts. Because they are not dependent on the cell division, and they have their own genetic material and are self-replicating, independent of the rest of the cell. So these have own genetic material and are self-replicating, independent of the rest of the cell. And then the other exception is the viruses, which have many features of life, but they're unable to reproduce on their own. So unable to reproduce on their own. and they have to be inside a host cell in order to be able to introduce. So therefore, since these viruses are unable to reproduce on their own, they are not considered as a living organism. And then referring back to the processes of evolution, evolution actually says that we have all developed from simple cells that later on became multicellular, and no one exactly can tell what had happened because we can't go back in time, but there are hypotheses about how different cells have developed. So first let me go over the definition of what prokaryote mean and what eukaryote mean. Okay, so first starting with prokaryote, which are thought to be the very first cells that have developed, and later on it's thought that eukaryote have developed from prokaryotic cells. So what prokaryote basically mean is that pro means before and karyote means nucleus. So it's before a nucleus has developed. In other words, prokaryotes don't have a nucleus, versus eukaryotic cells, they do have a nucleus, and the meaning of eukaryote is true nucleus. And then the theory is about how chloroplasts and mitochondria have developed in the eukaryotic cells. It's thought that when there was an anaerobic eukaryotic cell that was not using oxygen for energy production, it must have engulfed a prokaryotic cell that was aerobic. It was using oxygen to produce energy. And over time, this prokaryotic cell has been converted into mitochondrion, which uses oxygen for energy production. And then later on, this primitive eukaryotic cells picked up another prokaryotic cell that is able to use photosynthesis in order to generate carbohydrates from light and CO2 and water. And so that's how plant cells have developed. Now, there are many different findings that keep supporting the fact that chloroplasts and mitochondria were probably primitive prokaryotic cells that have been engulfed by eukaryotic cells. Like for instance, the size of chloroplasts and mitochondria is the same as prokaryotic cells. They have the same ribosome structure. They have their own DNA. So there are many different features of these chloroplasts and mitochondria that support the fact that eukaryotic cells probably engulf these prokaryotes, and over time they turn into the organelles that are used for energy production as mitochondria, or production of sugar and other carbohydrates, like for instance glucose, from sunlight that combines CO2 with water. All right, now moving to classification of cells. So cells can be classified into two main groups. One is prokaryotic cells, which includes archaea and bacteria. And then the other one is eukaryotic cells, which include animal cells, plant cell, fungi, and protists. So here you can see that the size of a typical eukaryotic cells is about 10,000 nanometer. The nucleus of eukaryotic cell is about 2,000 to 3,000 nanometer. And then a bacteria, which is a prokaryotic cell, is about 2,000 nanometers. So bacteria are much smaller than eukaryotic cells. And then comparing the size of bacteria and eukaryotic cells to viruses, you can see that viruses are much smaller, and then prions are even smaller. And then, as I just discussed in the next video, we will go over viruses, prions, and viroids. Basically, these are non-cellular infectious particles that will multiply only when they are inside the cell. So in the next video, we will go over viruses, prions, and viroids and discuss how these non-living particles are able to replicate once they are inside a living cell. Okay, now moving on with prokaryotes and eukaryotes. So in comparison to eukaryotes, prokaryotes are smaller, simpler, and lack a nucleus or any membrane-bound organelles. So membrane-bound organelles, like for instance mitochondria, are absent. Mitochondria, peroxisomes, these are all absent in the prokaryotic cells. They're made only of a single loop of chromosome and there are no histones in prokaryotic cells. So here you can see that the DNA of prokaryotic cells is circular and it doesn't contain any histones. Versus eukaryotic cells, it has a linear DNA. So the DNA in eukaryotic cells is linear. It uses histones to combine them with the DNA, and combination of DNA with histone is called a nucleosome. And so nucleosomes are strings of beads that will condense and help form chromatin. So that's the way that eukaryotic cells create a compact form of their DNA. The other difference is that during their replication, prokaryotic DNA has only a single origin of replication that will go all around and create two separate circular DNA. Versus eukaryotic cells, they have multiple origins of replication. So they start from multiple areas and start replicating DNA. So going back to the top, so eukaryotic cells, of course, they're larger, more complex than prokaryotic cells. They do have nucleus. They do have membrane-bound organelles. Their DNA is linear. So linear DNA that contains histones. And then eukaryotic cells divide by mitosis or by meiosis, versus prokaryotic cells that divide by binary fission. All right, now moving on. Here you can see the compact structure of a DNA in a eukaryotic cells. As you can see, DNA is being wrapped around histones in order to form nucleosomes. And then there is a string of nucleosomes that resembles a string of beads that compact into chromatin. And then chromatin during replication further compacts to form chromosomes. So that's what happens during mitosis or meiosis. Chromatin becomes more condensed and form chromosomes. So here I've provided you with a table that summarizes the differences between prokaryotic cells and eukaryotic cells. So prokaryotic cells, they have only one circular chromosome and is not membrane bound. So the chromosome is not inside a nucleus. Versus eukaryotic cells, they can have one or many chromosomes. Like for instance, humans have 46 chromosomes. And these chromosomes are bounded by a nuclear membrane. So there is these chromosomes are located within nucleus, and there is a membrane around it, which is called nuclear membrane. In terms of organization of genetic material, there are no histones or no introns in bacteria. So histones, we just discussed, are in eukaryotic cells in order to help form compact form of DNA. And then in our replication, in our mitosis and meiosis video, we just discussed how after mRNA is transcribed from DNA, eukaryotic cells have segments of mRNA that are non-coding. And those non-coding segments are called introns that needs to be coming out throughout the splicing process. But introns are only found in eukaryotic cells. There are no introns in prokaryotic cells. The next difference is the absence of any membrane-bound organelles in prokaryotic cells. So for instance, you know how eukaryotic cells have nucleus, lysosome, mitochondria, and peroxisome, all of which has a membrane around it. That feature is only present in eukaryotic cells. Bacteria, anything that is inside the confine of cells is not membrane bound. All right, and then finally, bacteria can have peptidoglycan wall versus eukaryotic cells that can't have peptidoglycan wall. We discussed how cell division in bacteria is by binary fission versus in eukaryote it's by mitosis or meiosis. DNA origin of replication, it's single in bacteria versus multiple origins of replication in eukaryotes. And so this is again what I'm referring to. In bacteria, DNA replication starts from one point and goes all the way around from both sides. Versus in prokaryotic cells, multiple origins of replication initiates and then starts spreading to different sides. And then here I have an image that summarizes the differences between eukaryotes and prokaryotes. So both eukaryotes and prokaryotes have a cell membrane that maintains the content of these cells. they both have ribosomes that are required for production of protein, but there is difference in the structure of prokaryotes and eukaryotes ribosomes. So eukaryotes are made of a 40S ribosomal subunit combined with a 60S ribosomal subunit, and then this combination refers to an 80S ribosome. That's what eukaryotes have. Versus prokaryotes, they have a 30S subunit combined with a 50S subunit to yield the 70S ribosomes. Alright, so here I have provided you with an image that shows the difference between eukaryotic and prokaryotic ribosome. So you can see that there are 60S and 40S ribosomal subunits, and there is ribosomal RNA that is inside these subunits. Their combination will yield an 80S ribosome, versus in prokaryotic ribosomes, the 50S and 30S subunits, which also have their own ribosomal RNA inside, they would combine to yield 70S ribosome structure. So the structure of ribosomes in prokaryotic cells is different than eukaryotic cells, and that's actually one method of designing antibiotics, that is to target prokaryotic ribosome, because that antibiotic that interact with either the 50S or the 30S subunit, it doesn't interact usually with the 60S or 40S of eukaryotic cells because it's a different structure. And so that's one way of differentiating prokaryotes from eukaryotes during the antibiotic treatment. Now let's move on to different classification of prokaryotes. So as we just discussed, there are two types of prokaryotes. One is archaea and second one is bacteria. So archaea has some features of eukaryotic cells. For instance, its genetic material is similar to eukaryotic cells, and they contain, for instance, histones, and they contain introns, and similar to eukaryotic cells, they lack the peptidoglycan cell wall. But then the reason they are being classified as prokaryote is that, similar to bacteria, there is no membrane bound organelles within the archaea. And then an interesting feature of archaea is that these prokaryotes produce certain cellular proteins that helps them survive under extreme conditions, like for instance under extreme heat, like you could find them inside boiling hot springs, or these archaea survive extreme salt concentration in the surrounding environment, and again you can find them in salty lakes. So these archaea, they produce proteins that helps them adapt to extreme situations. But then I would like to focus on bacteria, and here what I'm going to do is that I will go over the classification. So there are different modes of classification for bacteria. I will discuss different I would describe those classifications with you. Then I would go over the structure of bacteria, describe how there are different modes that a gene can be transferred to different bacteria, describe the mechanism of reproduction in bacteria, which is binary fission, and then finally discuss symbiosis, which is living of two different species together. Alright, so now in terms of classification of bacteria, as we just discussed, there are different ways of classifying bacteria. We can classify them based on their shape, we can classify them based on whether they are aerobic or anaerobic, and we can classify them based on the gram stain. So let's go over different one of these classifications. So the first one is shape. Bacteria can basically exist in three common shapes. And these structures, I've actually shown it to you right here. The first one is a spherical shape, which is called coccus. So coccus or spherical shape. The second one is rod or bacillus. So here we have rod or bacillus. Okay, rod shaped or bacillus. And then finally, the third one, which is these two that is shown right here, is spiral shaped, which could be in the form of a spirochete, which is very long and flexible, or it could be in the form of a spiralome, which is short and rigid. So the next form is spiral shaped. The next mode of classification is aerobic versus anaerobic. So anaerobic are, I should say strict anaerobic, are bacteria that are not using oxygen. And an example of a strict anaerobic is, for instance, the Prokaryotes that are found in the large intestine, versus aerobic are organisms that are using oxygen for energy production. And then we have another form that is called facultative anaerobes. And these are basically a more versatile form of bacteria that can grow under both high and low concentration of oxygen. So keep in mind, facultative anaerobes can grow under both high and low concentration of oxygen, versus strict anaerobes, they have to grow under conditions that there is no oxygen. And strict aerobes, they require oxygen for their growth. Now the next mechanism of classification is Gram stain. Basically this stain was discovered in the 19th century, and it basically uses different compounds in order to stain the bacteria. The ones that are colored blue or purple, these are called gram positive bacteria, and the ones that are stained pink or red are called gram negative bacteria. So let me go over different components that are being used for the gram staining. So first, a crystal violet, which is a purple colored dye, is added. And this crystal violet, along with combination with the iodine, which increases the affinity of the stain for cells, is staining the peptidoglycan cell wall of bacteria. In the next step, all the dye is being washed off. So the bacteria that have a thick cell wall, which are gram positive, will retain the crystal violet color, the purple or bluish color, versus the ones that are gram negative and have a very thin wall will lose the stain. And then in the final step, we are using a safranin, which stains gram negative bacteria a pink color. Okay, so again here you can see the difference between cell wall structure of gram positive versus gram negative bacteria. So gram negative bacteria have 2 layers of cell membrane and a small peptidoglycan cell wall that is located in between, versus gram positive bacteria have a very thick cell wall and only a single plasma membrane. The other difference between gram positive and gram negative bacteria is that gram positive bacteria have teichoic acid, while gram negative have lipopolysaccharides on their cell membrane. And the way I memorize it is that I always tell that teichoic acid, T looks like a plus, teichoic acid. So that's how I remember that there is teichoic acid in gram positive cells versus there is lipopolysaccharides in gram negative cells. Now you can see how the peptidoglycan cell wall is very thick in gram positive bacteria. Again, the way I memorize it is that I think of the plus sign like this. Their peptidoglycan cell wall is very thick, versus gram negative bacteria that have a thin wall. And that's the basis for the difference in the staining pattern of gram positive versus gram negative bacteria. Gram positive bacteria have a thick wall, and therefore crystal violet stain with the help of iodine will remain there even after we wash the cells with alcohol, versus gram negative have a very small cell wall, very thin cell wall. So the crystal violet color will be lost, but we need to somehow visualize these cells. So that's why we add the safranin contour stain, which gives these cells a reddish color. And so again, gram negative has a thin peptidoglycan wall and won't be able to retain the crystal violet color. Now, please note that there is also a space in between the 2 membranes, the outer membrane and inner membrane of gram negative bacteria, and there are some proteins that can be staying here, and these proteins sometimes help gram negative bacteria in order to deactivate the antibiotics that we are using against them. And then as for lipopolysaccharides that are found on gram negative bacteria, the other name for these lipopolysaccharides is endotoxin. So the function of these lipopolysaccharides is to provide integrity and stabilization to the outer membrane of gram-negative bacteria. But the problem is that inside the host cells, like human cells, these endotoxins will initiate a severe inflammatory reaction. And that's why the name endotoxin is given to these lipopolysaccharides. All right. Now we are done with the classification of the prokaryotes. Let's move on to different structures that exist in prokaryotes. And so basically, the first structure I would like to discuss is the cell envelope, which is composed of a cell membrane surrounding the internal structures of the cell. Then we have a cell wall, and then finally the green layer that is shown right here is the capsule. So let's go over every one of these structures. And these are all classified under classification of cell envelope. So regarding the plasma membrane, this is actually a lipid bilayer that is mixed with proteins in order to form a fluid mixture. So this is a lipid bilayer, and it helps regulate trafficking of molecules into and out of cytoplasm. So it regulates trafficking of molecules. Next, we have the cell wall, and this is basically a cross-linked layers of peptidoglycan. So, you know how we were talking about the peptidoglycan cell wall in gram-positive that is thicker versus gram-negative here. So that's actually the structure we are talking about, the cell wall. And basically the job of the cell wall to help maintain the shape of the bacterial cells. So it maintains shape of bacterial cells. And then finally we have the capsule. Let me come down a little bit. And finally we have the capsule, which its composition is made of a glycocalyx, which is a thin layer of polysaccharide polymers, or in other words, basically it's a sugar coat that lies outside the cell. And this capsule makes bacteria very resistant. It protects them against drying and then also against phagocytosis by different immune cells. All right. Then the next component of prokaryotes is cytoplasm, which is basically a semi-fluid solution that is encased by the plasma membrane. And the structures that you will find inside prokaryotic cells include a nucleoid. So nucleoid is basically the region that contains the coiled circular DNA. So you know how we just discussed in bacterial cells, the circular DNA or circular chromosome is not having any membrane, that is not having any nuclear membrane. But the region inside the bacteria that you find the circular DNA, this coiled circular DNA, it's referred to as nucleoid. Then bacterial cells could also have plasmids in their cytoplasm, and basically plasmids, like as shown here, these are small DNA structures that are circular and are separate from the actual genomic material of the bacteria. And the function of these plasmids is usually to provide antibiotic resistance. They can produce different toxins, and they could also help with synthesis of different proteins or enzymes that helps with the survival of the prokaryotic cells. Okay, and these plasmids, as I will discuss later on in this video, can be transferred from one bacterium to another. So that's why if you combine a bacteria, a strain that is resistant to certain antibiotics with another one that is sensitive, you can sometimes develop resistance, and then you're like, what happened? So in those cases, it's usually due to the transfer of plasmid from the bacterial strain that was resistant, and there was like a protein or something that on that plasmid that breaks down the antibiotic. So by transferring this plasmid to the other strain that was previously insensitive, there would be resistance development. All right, now the next structure are the inclusion bodies, and these are basically nutrient granules that are used in times that there is a low source of energy available. So these are nutrient granules. Next, we have the ribosomes, and we've already discussed the ribosomes. Basically, these are composed of a large and a small subunit. So here in prokaryotes, we have a 50S large subunit and a 30S small subunit. And the function of ribosomes is to produce proteins during the translation process. So you know how DNA can be transcribed into mRNA, and then mRNA is translated into protein. That's the job of ribosome right here, with the help of the tRNA to form proteins from the code that is inside the mRNA. The next structure is the appendages, which are actually protein structures that can assist in various different functions. And these include, for instance, flagella, fimea, and sex pili. So let's first focus on flagella. So basically flagella, it's made of a chain of a protein called flagellin that anchors to the cell wall. So here you have the flagellin that anchors to the cell wall, and its function is to help the bacteria to move around. So you might recall from eukaryotic lecture that we discussed a sperm that has flagellum. There's a bending motion to the sperm flagellum, and it's kind of like rippling motion in order to help the sperm propel forward. In prokaryotes, which we are talking right now, it's a completely different mechanism. It's a more simple structure, and it can't really move in a bending motion. So there is two methods of regulating the motion of, or the movement of the bacteria, and that is whether these flagella are being rotated counterclockwise or clockwise. So in bacteria, we only have rotatory movement, versus in eukaryotes, like in the sperm, we had bending movement. So bacteria depend on rotatory movement. If there is a counterclockwise motility of the flagella, that's the time that it will push the bacteria forward. That's why sometimes the counterclockwise motility is referred to as running mode. While if there is a clockwise rotation of the flagellum, that's the time that it will make the bacterium to rotate around itself. So it will start to move around itself, and that's why it's sometimes referred to as a tumbling mode. Now, if a bacteria is being exposed to some chemicals that is being perceived as attractant to the bacterium, like for instance there is sugar and bacteria loves sugar, then they would increase their counterclockwise motility so that they would move towards that attractant. On the other hand, if they get exposed to some sort of chemicals that is perceived as repellent, like very high salt concentration, then they would increase the clockwise motility of their flagellum so that they would turn around and run away. So they would turn around, and then with the help of counterclockwise, they could move forward and move to a different direction. So here you can see that we have a bacterium that has its flagellum moving in a counterclockwise. So it would come forward until at this stage it decides to change the rotation direction and start moving its flagellum in a clockwise. So now it starts to tumble and move around, and then the new direction that it picks is this way. Again, it will have a counterclockwise motility, run, then tumbles, run, tumbles, run. So that's basically how bacteria can move around, either based on the counterclockwise or running mode of the flagellum, or clockwise motility of the flagellum, which initiates a tumbling mode. Now the next two appendages that I would like to talk about are fimea and sex pili. So basically fimea, these are shorter and thinner than flagella and are not used for motility, but instead they're used for attachment of bacteria to different surfaces. And then we have the sex pili, which are basically tubular structures that help transfer DNA from one bacterium to another bacterium, and this process refers to as conjugation when a pili is used. And that's basically the next topic that we are discussing, different modes of gene transfer. And so if a sex pili is used to help transfer a plasmid from one bacterium to another, this process is called conjugation. So let's now go over different modes of gene transfer from one bacterium to another. So the first one is called transformation, where one bacterium picks up any extracellular DNA that it finds in its environment, whether it's from another live or another dead bacteria. And the example that is shown right here is right here. So you can see that there is some DNA that is left over from this bacteria that is shown right here, and the other bacteria at the bottom picks up this DNA structure and incorporates it into its chromosome. So given that for this process the DNA has to be found in the extracellular structure, if you add DNAs to the culture medium, you can interfere with the transformation process. Okay, so just keep that in mind. Since DNA has to be released inside the extracellular environment in order to be picked up by the other cell, if you add DNAs, which is an enzyme that breaks down DNA, it would eat up any DNA that is out there, and so therefore transformation could be inhibited. All right, the next phase, the next mode of transferring gene is called transduction, and that's actually being induced by bacteriophages. So once bacteriophages infect one bacteria, if they go on and infect another one and they inserts their DNA into the DNA of the new bacteria that they infected, this is called transduction. So basically transduction is when a bacteriophage helps move around DNA from one bacterium to another. The next mechanism, as we just discussed, is conjugation, where the donor bacteria is able to transfer DNA to the recipient with the formation of the sex pili. And the bacteria that are able to form the sex pili, they're having the F plasmids. Those are called F plus bacteria, versus the ones that don't have it are called F minus bacteria. Okay, next let's go over the mode of reproduction in bacteria. We already discussed that bacteria are replicating by binary fission, or in other words, asexual reproduction. So here you're having binary fission or asexual reproduction, versus in eukaryotic cells that are dependent on mitosis or meiosis. So now let's focus on binary fission right here. Basically during this process, a two identical copy of circular chromosome or circular DNA has to be made, after which the cell will enlarge and divide in half to form two new daughter cells. So in the first phase there will have to be DNA replication. So here you can see that there is DNA replication. As we just discussed, it will start at a single origin of replication and it will go around until there would be two circular DNA that are formed. And then once the two circular DNAs are formed, the cell will undergo division, where the plasma membrane will invaginate and form a cleavage farrow, and then a septum will be formed, and then after which the parental cells separate into two identical daughter cells. So that's basically the process of binary fission. And then I would like to mention that the process of binary fission can happen very quickly. Actually, it can happen as fast as 10 minutes. And so in the presence of nutrients, that's why that the bacteria can grow very rapidly. However, we know that the nutritional sources are limited. There can't be unlimited nutritional sources. And that's why usually the growth phase of bacterium, this is how you will see. There would be a lag phase that the bacteria is not growing. Once there is exposure to nutrients, there is an exponential growth phase, or the log phase, so referred to as log or exponential growth phase. Now at this stage, the nutrients are starting to become less and less, so there would be a plateau or the stationary phase, and then finally the toxins and the byproducts will build up, or in other words, the waste material will build up that will make the bacteria to die. And so that would be the death phase that is happening right there. All right, and then for the final part of this video, I would like to focus on the difference in the reproduction of eukaryotes versus prokaryotes. So you know how in eukaryotic cells, they can divide by either mitosis like regular somatic cells, and that a 2N diploid cells will yield another 2N diploid cell. And I should have said a 2N diploid cell gives rise to two different 2N diploid cells. Versus in meiosis process, where you see in germline cells, a 2N germline cell gives rise to 4 different haploid cells. And these are sperm, or if you're talking about the female ovaries, it would be an egg. And then I'd like to mention that female ovaries, there would be disproportional distribution of cytoplasm. So usually 3 of these are polar bodies that will degenerate. So this is all discussed in the reproductive video. My point is that during the meiosis, there would be crossover between chromosomes, as a consequence of which there would be variation in the type of cells that are being generated. They're not being identical. In bacterial cells, however, there is no crossover. So that's what I would like to discuss next, and that is how can bacteria develop some genetic variation, or in other words, genetic adaptability, in order to survive different environments. So basically, there are different ways that bacteria can have genetic adaptability, or in other words, genetic variation, and these include, for instance, horizontal gene transfer techniques, which includes, for instance, transformation, conjugation, and transduction. So these methods of gene transfer that we just discussed, these are horizontal gene transfer that gives variation to the bacterial genome. Other modes of developing more genetic variation is via random mutations that happens in the genome of bacteria. Okay, and it helps these bacteria to adapt to new environments. And then finally, genetic material can move around in the genome with the help of transposon elements. And so the phenomenon of having genetic material move from one point to another is called transposition. And so next I would like to emphasize how any one of these methods of genetic variation can help with the survival of bacteria. So for instance, horizontal gene transfer, as we just discussed, there is a plasmid that has antibiotic resistance gene, or there's a piece of DNA, and so via the horizontal gene transfer, one bacterium can pick up this resistance gene, and so therefore fight off the antibiotic that is trying to kill it. So that's one way of helping with the survival of the bacteria. Alternatively, the bacterium can develop mutations, can undergo random mutations that will change its structural components that would not be any longer sensitive to that specific antibiotic. And then finally, transposons. So imagine if you have a gene that undergoes duplication, and then transposon moves this gene somewhere else in the genome, and over years this new gene undergoes new mutations and undertake a new function that helps with the survival of the bacteria. So all these mechanisms increase the genetic adaptability of bacteria and help them with the survival. Alright, next I would like to discuss the definition of symbiosis, and that's the last topic that we are discussing in this lecture, and then we will continue the rest of the topics in our second video. Alright, so symbiosis, this is basically defined as having two different species living together. And whether they harm each other or they benefit each other, that's when we can come up with different terminologies. So if there's a relationship between the two different individuals and both individuals benefit from these relationships, then this is called a mutualism. An example is the bacteria that is found in our digestive tract. So these bacteria will use up the nutrients that we are eating, but then in return they are producing vitamin K for us. So this is a mutualism relationship. We both benefit from living together. The other relationship is commensalism, where one species benefits while the other one is neither harmed nor is benefited. Like for instance, once the plant seeds stick to animal fur, they do get benefited by the fact that they can now go to a different environment and start a new plant there. But then the animal is neither harmed nor helped. And then finally, we have parasitism, where one organism benefits while the other one is being harmed. Like for instance, the worms that are in our GI tract, or sexually transmitted diseases. They found a new home to grow, but then they caused trouble for their host. All right, so that would be an example of parasitism. All right, so basically that concludes the discussion of the first part of this video. I'm going to end this lecture, and then we will continue on the rest of the topics that are shown right here in the second part of this lecture. So thank you all for listening, and see you for the next video.